The driver for digitizing pathology was not super strong. And then we saw these AI applications starting to come out on the first sets of digitized pathology images at a research level first. And I think that this opened up so many possibilities in everybody's mind. And then this basically in a kind of chicken and egg kind of virtuous cycle way started accelerating the digitization of pathology, which in turn increased the research capabilities and the amount of data science that we could do, which increased the proof points that we could do a lot with these images, which facilitated the investments on digitizing of pathology.
Hey everybody, this week I am joined by Meriem Sefta, PhD, Chief Diagnostics Officer at Owkin. Owkin is the first end-to-end AI biology company on a mission to understand complex biology and make sure that every patient gets the right treatment using AI trained on world-class patient data through privacy-enhancing technologies. So, Meriem, delighted to have you. Welcome. How are you doing?
I'm doing good. Thank you for having me.
You're very welcome. Whereabouts are you speaking to us from today, Meriem? Where are you based?
I am based in Paris, France.
Oh, very nice. Very nice. Yes, because we had the we had the building problem last time we tried to record, didn't we?
Yes.
Yes.
Construction in the building. That was fun.
Yeah, it's everyone taking down everything from the Olympics, isn't it? Like causing a lot of noise.
There was massive construction for 2 years, I would say, before the Olympics.
Oh, wow. What's it been like?
The Olympics was a lot of fun. Actually,
Yeah.
Parisians tend to be pretty pessimistic, so there were kind of a lot of bad projections before the Olympics.
Okay.
People thought it was going to be too crowded, not well organized, not fun, and in the end, everything went well. It was a lot of fun.
Yeah. The opening ceremony took a bit of heat, but I think beyond that, it all seems to have gone quite swimmingly. So, yeah, because I can remember when we had it in London, it was just glorious just being in London, like with everything going on. It just felt like the place to be. It was so exciting. But, yeah, and obviously Paris being such a lovely city anyway, I imagine it's been quite a fun summer.
Yeah, it was.
Cool. So rather than talking about Olympics, although I could probably talk about sport for another hour and a half, let's talk about some biotech. So I guess for those people that don't know, I mean, everything that I've read and seen about Owkin is fascinating. And it's interesting, even in the introduction, you specifically call out, world-class patient data and that almost being like a differentiator. And I think it's such an important thing to get right, especially when there's so much call for, diverse data sets and everything to that. But I'm sure we'll get into all of this because you've got a fascinating title as well, Chief Diagnostics Officer. And so I think learning what a Chief Diagnostics Officer does and what that part of the business is responsible for and looks like will be fascinating. But before we get into it, it'd be great to hear your story. So I guess, how does someone go about a career to become a PhD and a Chief Diagnostics Officer at a biotech company like Owkin? Why don't you tell us the whole story?
So I actually am an engineer by training. So basically, I grew up in the U.S., but I wanted to study in France. And at the end of high school, basically, I considered med school. But doctors typically train in a specific country in a specific language, and I didn't really like that because I didn't want to get committed to a particular country for the rest of my life. And so I went to engineering because I figured, well, you can do engineering wherever. But then during my engineering studies, I kind of started leaning towards everything that had to do with biomedical, just because I think it's something that always attracted me. And just all the ways that in engineering through engineering, you can basically help medical use cases. And so then I did, the kind of the first 5 years I did in France. And then I moved to Boston in the US to go to MIT. And I was in the biological engineering department. And there I did wet lab, so kind of experimental biology, just enough to realize that it was not my thing. Yeah, I think it takes a lot of dedication and precision and patience, which is not me, I would say, in terms of precision and patience. And so then I switched to computational biology, which I liked a lot more. So really completely computer-based research. And I did my PhD in that, for which I moved back to France, and I was at the so still, basically from the beginning, I kind of went towards everything that had to do with oncology. The reason for that is that I just think that it's the complexity of what is going on in a cancer is very, I would say, interesting and exciting to study when you're an engineer. Basically, it's a very big research challenge. And so then I moved back to France, and I was at the Curie Institute for Cancer Research, basically, in Paris. And the way that the Curie Institute works is that it's a hospital and a research center. So kind of two branches to the institute. So you have basically patients being treated by care professionals in the hospital part, and then a whole research center that works on basically cancer biology research, and they collaborate with people from the hospital and clinicians from the hospital. So there's a very fluid, I would say, collaboration between both, and I think that's really one of the biggest strengths of this institute, is that there's a direct connection between the researchers and the clinicians.
That's such a good point, by the way, just to jump in, because I think this is something that, do you know what, ever since I've been in, I think, the startup world full stop, I've just noticed this phenomenon so much where two groups that don't normally collide, that's sort of where new ideas live. And so I've seen it across health tech, definitely. The more sort of clinicians that become engineers and computer scientists, all of a sudden their brain works in two completely different ways that can not only see a problem, but also see a solution. And I can remember when this is years ago, I went to Finland and got invited there because of that Microsoft were doing some thing in Finland. And it's the same thing in Helsinki. They've got, in their hospital, they've obviously got the hospital function, but then they've got an innovation function that just lives inside the hospital. So you get in the corridor, like doctors and computer scientists bumping into each other, or students of medicine and computer science and all the rest of it bumping into each other. And just as a very sort of physical and very human factors, I guess, like chaos theory almost. There's just so many new ideas that live there, and so many extra things get done. It's fascinating that where these things are actually engineered, that you get the new ideas that that you're talking about.
Yeah, exactly. And especially for doctors are first, they're kind of, I would say, one of the current professions that is still not at all digital. They don't spend their days on their computer, on Zoom meetings. They're in their hospital treating patients, moving from one room to the next, from one floor to the next. And time is the number one thing that they are lacking. And so I would say the most efficient way of working with them is to be as physically present as possible, to catch 5 minutes, 10 minutes, walk with them in a hallway, rapid fire questions. And that's how you get as much information as possible, and how you can be as efficient as possible in collaborating with doctors, basically.
Yeah, they will massively appreciate you saying that as well, because that's so respectful of the only resource they have, which is time.
Yeah.
Or that they don't have, I should say.
That they're lacking of.
Yeah, definitely.
Time.
Definitely.
It's a really question of not wasting their time and just making the most of the time that you get with them to be as efficient as possible, and also to get a kind of sometimes I think, especially when you're an engineer and you're working on data, data science in other fields, the data is very structured. There's no lack of data. If we're talking about applications such as, looking at images of cats and dogs, or training on data that you scrap from the internet, you don't lack data, and you don't lack data ability to structure the data. And the thing about data from a hospital is that it's, for lack of a better word, a mess. There are different information systems that are in different parts of the hospital, different softwares. It's difficult to extract the data. It's difficult to structure it. There are a lot of initiatives in all the hospitals to improve this, but it's still a lot of work and a lot of time that is required to do this. And when you're an engineer and you're used to kind of, I would say, better structured data in other fields, it's very hard to grasp what environment these people are working in and what their hurdles are, unless you actually see it for yourself. You see the actual computers that they're working on, the actual, software that they have, the way the information flows in the hospital, and then you understand kind of at what level of, data readiness you're working with, and what you need to do to actually be able to get, the information, the data quality that you need to do, whatever data science or engineering objective and project you have. And I think that as an engineer, if you haven't actually, looked with them at the data, it's I think it's very difficult to grasp.
Yeah, it's a great opening gambit to your story, because I think it speaks to a concept that we definitely need in biotech and health tech, and just even in medicine, which is listening first. I think people often forget this as well. I think it's very, very easy for people in their silos, forgive the jargon, but it's easy for people to just think, they should do it this way, they should do it that way. I think it's easy to learn engineering and computer science concepts and then to think that, they can and should be applied. It's a very easy mindset to be in, because I'm in the same mindset. Often the longer I I've been stepped out of medicine, the more I'm just, my frustration of just, why can't you just do this or just do that, starts to creep in, and then I have to sort of kick my empathy back into gear of the actual situation that people are in. But it's really nice. Like, what you're talking about is just listening first. You're genuinely just talking about the early part of your career, just actually just sitting and listening to people, which is wonderful. So yeah, you definitely have a captive audience, I think, of the clinicians that are listening.
Cool. All right. Well, and so that's where I did my PhD. And also, I would say one of the things that really drove me to understanding how data was structured in a hospital and how we could work on data was that for my PhD, I had a bioinformatics project on a rare cancer called retinoblastoma. And the Curie Institute is a national reference center for retinoblastoma, which means that all patients in France get referred to the Curie Institute as soon as a retinoblastoma is highly suspected or diagnosed.
And just people that don't know, what is a retinoblastoma, just broadly?
Oh, okay. So it's basically, retinoblastoma is a pediatric cancer of the retina. It affects very young children. Sometimes it's diagnosed at birth. It's highly treatable. So I would say the prognosis in terms of survival is very good, almost close to 100% in developed countries. I would say that the kind of key challenges for retinoblastoma care is, one, so it affects the retina, and so it's really a question of preserving the eye and the eyesight of the affected eye or eyes, because sometimes it's bilateral, so in both eyes. And the second is also working with developing countries to help them diagnose and treat this cancer early enough that the prognosis, the survival prognosis reaches the level that it's at in the more wealthy countries, basically.
And so what was your PhD on specifically?
So my PhD was on genomic analysis of basically building a genomic profile of these retinoblastoma cancers. So this was I started my PhD in 2012. And back then, kind of the new thing that all the researchers were looking into in oncology was genomics, DNA sequencing, RNA sequencing, looking at the mutations that are driving these cancers, subtyping and subgrouping the cancers, because through basically the ability to sequence the human genome, the transcriptome, and generate all of this omic data basically on cancer tumors. These researchers were more and more identifying that, when you speak of cancer, you're actually, for example, of breast cancer, you're actually looking at a family of diseases with different subgroups of patients that have actually sometimes very different molecular profiles and then also clinical profiles. And so it was kind of in that era where people were now looking at the molecular profiles and matching that to kind of different clinical outcomes and different clinical profiles of these patients. So kind of really opening the box and seeing what was inside at the molecular level. This had been done, I would say, already for a few years for kind of the more prevalent cancers, such as, for example, breast cancer, which was one of the first ones. Before rarer cancers such as retinoblastoma, nobody had actually looked. And so we were among the first to really sequence the genome of retinoblastomas, understand what was going on, see if there were particular subgroups, particular different genetic drivers in these tumors, were there, patient subgroups, things like that. So that was kind of the story of my PhD. And I was doing this in I was co-advised. So I had a biologist and a computational researcher advising me. And so we were kind of going back and forth between the computational biology, and the analysis of this genomic data, the clinical data in the hospital. So really understanding the clinical profiles of the patients and, does any heterogeneity that we see at the molecular level match what we see at the clinical level? And then also at the experimental level, we're working on cell lines and models to model these tumors and see if, from an experimental perspective, we could validate some of our hypotheses on what was driving these different, for example, cancer subgroups.
Wow. Broadly, what did you find?
Broadly, we found that retinoblastoma, first and foremost, like a lot of pediatric cancers, actually has a very non-altered genome. So in adult cancer, so this is the kind of mystery of pediatric cancers. Yeah. Adult cancers, it's basically the accumulation of DNA damage that eventually leads to cancer.
Yeah.
And so when you look at an adult tumor, you see so much damage. Yeah. And it's kind of you're basically seeing kind of the archives of years and years of damage that accumulate and eventually create a cancer. Right. But, how do you explain? And so it becomes logical that the older you get, the more likely you are to get cancer. Right. Then how do you explain pediatric cancers? And they basically answer a different set of rules. The genome is actually not very much altered. But it's kind of at phases of development where there's such plasticity in the cells that sometimes one alteration can be enough to drive something kind of off the rails, basically, because it's a moment where, everything is growing, is developing, there are growth hormones going on, there are changes, lots of plasticity. And so, nothing is locked. An adult kind of cell, you really have to hit it a lot of times before something changes, because everything is kind of stable and locked down. Whereas,
and you have an immune system that's more fully functioned, I imagine, just taking out the stuff that's
I don't know, but I imagine as well. Whereas for pediatric cancers, everything is moving around, everything is very sensitive, everything is growing all the time, evolving. Wow. And so it's kind of like situations where if you hit the wrong thing at the wrong moment, you can basically drive a tumor. And so the question is, what are those hits? What are those alterations that are driving these pediatric cancers? Are they the same for all the patients? Are there some? And so we identified, for example, one driver alteration that had not been as extensively studied, and we showed that it was a subgroup of patients that were really driven by this alteration in these retinoblastoma patients, basically. So that was kind of one of those findings. And it, like I say this in a one sentence summary, but this was years of work of myself and other colleagues and clinicians and biologists that were also validating this work. And this was published, I think, maybe 4 years after I graduated. So really, it is a long endeavor. And one of the longest parts was actually getting the data. And so to circle back, because I talk a lot, but one of the reasons why I got so interested in data and how data was structured in a hospital was that because this was a rare cancer, my thesis advisor, his number one advice was every patient counts, every data point counts. This is not like a very prevalent cancer where you can afford to lose one patient on one data point. You only have so many patients a year, so you need to get all the data on these patients, and you can't afford to basically drop one or drop a data point. And so I actually had like a list of obviously anonymized patient IDs, and every single kind of not available data point or every single missing data point, I would kind of go hunt in the hospital to see if I could get that information to basically have a complete data set. We were only talking about 150 patients. It was really not that much. A lot for retinoblastoma because it's a very rare cancer, but not that many patients. And so through that, I kind of started more and more seeing how the data was structured in the hospital or not structured, how the data was captured or not captured. And that kind of gave me a better understanding of, I would say, what the pain points there were, what the challenges, and how to better work with and help these basically healthcare structures to structure their data.
That's incredible. I'm interested in before we just move on to Owkin, when you were looking at those hospitals and with the fresh eyes of someone that has come in, you can understand and appreciate how data should be structured or could be structured for maximum, I guess, harvesting and impact and all the rest of it. What was your sort of overwhelming feelings like when you went into that hospital and you were seeing it? Was it frustration? Is it like waste? Is it I don't know, like what were you feeling when you were walking around the hospital, and where do you think we could be with data based on what you know of what's possible?
So I think as an engineer, and when I started at Owkin and started working with so Owkin is just a lot of engineers working on medical applications, and we now also have doctor colleagues and biologists, but at the beginning was data scientists. And at the beginning everybody was kind of thinking the same thing. It is frustration that I would say the information is there. It's just not structured and not consolidated in a way that it is easily queryable and analyzable. And how do you say that? You can easily query it and analyze it. And so when you're a data scientist, you can see all these potential use cases, all these potential research questions, and you're collaborating with doctors and you're discussing all these things, and then you hit always the same hurdle, which is, but the data is a little bit everywhere and we don't know how to get it. And then the second frustration is, I would say, lack of time and money from these healthcare systems to actually put the data together. As an engineer, it kind of drives you nuts because you're like, this is not rocket science. This is pretty simple stuff. This shouldn't be what is holding us back. And yet it is.
No, it is. Because it's funny because my frustration, I can remember being in ITU and thinking this like way back in the day. that my frustration definitely around structure and the ability to query and all that sort of stuff. Although I wasn't never a data scientist or engineer, so my thinking was never this structured. But what I tended to feel that there was also a problem with capture. Like we weren't there was so much data that was just sort of disappearing into the ether of how people looked or what they sounded like, or like, yes, okay, we put an arterial line in and we can get a a pressure trace, and we can get, temperature, and we are capturing some stuff. But I was thinking, like, God, imagine if we captured literally everything. But then then that, really multiplies the structure problem if you start doing that. So it's interesting the way you talk about it, that actually structure is the first thing to solve. And then once we solve that, we can think about potentially more capture and putting more data in. But yeah, fascinating.
Your point is very good as well. Yeah, I think there is also a question of just capturing more in a way that's that is easily usable down the line. Now, I say this and, I say frustration, but all of these systems were built for care, not as much
Absolutely.
for research. And so, and when people built these kind of healthcare systems and data capture and all these things, it was with the information of what was important or what mattered for care that they had at the time. So it wasn't, as looking back, you're like, why didn't they write this down, and why don't we have this information? But they were going off of, what they thought, what they knew was important. It's also, I remember we also did sidetrack. We did a project at Owkin on COVID in 2020, I believe, and we worked with Kremlin-Bicêtre APHP Hospital on basically all of their COVID cases. So APHP is a network of Paris hospitals, and it has I think it sees 10 million patients a year. So the volume of COVID cases that they had was really big. And so we looked at kind of, I think, one of the APHP centers and several of the APHP centers and their COVID patients. And at the height of COVID, if you look at the kind of ER logs of COVID patients, they were bringing in basically one every 15 minutes. Every 15 minutes, somebody would walk in, with respiratory distress. So you're just imagining the days that these ER doctors were living when you look at then the kind of the health information data that was input, and every 15 minutes they had somebody coming in with basically COVID kind of respiratory distress issue, and we kind of collected the data to analyze it. And so we kind of started structuring. We actually worked manually structuring the data because it was the fastest thing that we could do. So taking all the information that was logged in by these care providers across all of these different patients to analyze it, what you could see was that the data week 4 was so much more rich and so much more structured than week 1. Because week 1, they had no idea what they were actually looking at and faced with. So they didn't know what to capture or what to write down. And it was, it was just kind of notes scribbled down on the patient files. And then as the weeks went forward and, research was moving super fast at the time. And so more and more kind of information was flowing on, this is important. You need to measure this. You need to look at this. Blah, blah, blah. Over the course of just 4 or 5 weeks, the data started becoming more and more structured. Like, O2 sat, like a whole bunch of things that in the beginning was just like, I think they just didn't even know what to look at. And so I think it was kind of an accelerated view of how research and clinical understanding of a disease can progress. And it was accelerated over just a few weeks.
Yeah.
So,
I think what's really interesting about what you're saying is you're sort of talking to this new relationship between clinical medicine and clinical care and data. And we're moving from a time where the number one and only consideration of how you structured healthcare was the care part, because we didn't capture data if in any sort of, longitudinal fashion or anything of note for research once upon a time. And we're sort of in this transition phase now where we know and appreciate the value of data, but our systems are not quite there to appreciate capture. And I guess you're talking to this future, and this is where Owkin comes in, right? So we can talk about this as a segue into it, but you're talking about this future where the nature of clinical medicine has to, in part, be appreciative of the advancement of it through data. We have to appreciate that data is intrinsically linked to the future of healthcare being greater. And the more that we can actually appreciate the link between the two and build that into healthcare, then perhaps that's the key to a quicker and more innovative future. I think it's also useful to note, though, that there is always this tension in clinical innovation, I think, as well, because in order for anything to get more efficient, it needs to get less efficient first. And that's because everyone is at 100% capacity on the ground floor. And so to change anything, there has to always be this period of getting less efficient before you're more efficient. And very practically, that can look like bringing in a new computer system that is quicker. And it's like, well, we have to train our people on that. So where's that time coming from? It's coming from clinical care because everyone's at 100% capacity, or it's coming out of people's own personal time, or what the NHS particularly stands up on, which is the goodwill of everyone that works in it and that kind of thing. And so there is always this tension, but you're right. I think in order for us to appreciate and realize this greater future of healthcare that we all want and utilize the pace of what's going on in technology, the data capture and the data structure has to be built into the way that we practice medicine. And I can definitely see a world where those two things are much more linked, and the dual qualified people, or indeed even making it part of training in clinical medicine, the appreciation of data for our future. I don't know, but these are all musings of someone incredibly underqualified to be talking about them. So I will hand back over to you, Meriem, to talk about Owkin.
I think very few people are. I think it's so complex. Nobody's actually 100% qualified.
You're more qualified than me, though. So let's talk about Owkin. Let's talk about exactly what you do, and let's talk about the chief diagnostics role in the diagnostics department of Owkin and what you do and what you get up to.
So basically, So when I joined Owkin, I joined as first head of partnerships. So to head basically the data partnerships. So very similar to what I had done in the past of Owkin. So how do we collaborate with academics to build efficient research collaborations? Work with KOLs, but also work with hospital data to basically improve biomedical research. And I would say one of the data modalities that we worked with the most since the beginning of Owkin is digital pathology, so digitized pathology images. I would say one of the reasons for that was that at the time that we kind of started all of these collaborations, one of the most underexploited data modalities that existed was pathology slides. So pathology slides, I would say for those who don't know, are basically the bits of an oncology tumor, so either from surgical resections or biopsy, that are kind of sliced into tiny pieces and put on a glass slide and then observed by a pathologist under a microscope. And that gives them an idea of the morphology of the patient's tumor, and looking at the morphology, they can get a better grasp of what is going on, the level of aggressiveness of the cancer that they're looking at, even some features that, for example, would be predictive of prognosis, response to certain treatments, what is the best course of therapy that you can look at, that you can infer directly from these images, which are basically a molecular, a morphological cell level view of what is going on inside a patient's tumor. And for a very long time, these were basically glass slides that pathologists could look at under a microscope. And over the past 15 years, these glass slides have become more and more digitized. So slide scanners have come on the market, which allow you to generate just a digital image version of what a doctor can actually see under their microscope. And these images are actually very, very rich in information. So a full-sized digitized pathology image can be about 2 gigabytes of data.
Whoa.
Yeah, so that's about as much as a full-length feature film. So if you were to
I've not appreciated that before.
Yeah, it's huge. If you were to print out a digitized pathology image, it would be the kind of the whole side of the building. It's a huge image because you're really looking at the kind of tissue level and then really zooming down to really the cell level. And there's so many, millions of cells in just one, for example, tumor resection sample. And then you can also have multiple slides or multiple resections per patient. Talking about, for example, multiple biopsies or different parts of the same tumor that sometimes don't even look the same. So there's a lot of information in these slides, and until very recently, they were almost up to today, they were almost exclusively analyzed by pathologists visually. And a pathologist will typically have just a few minutes to look at a pathology slide. So that means that in a routine care setting, they need to basically quickly identify the key features that are in these huge images that they need to basically look at and identify for that are important, that we know are important for patient care. And at the beginning of Owkin and our data science and research team, we were among the first in the world to show that using AI on these images, we could basically further exploit kind of the wealth of information that was in these images through the AI and identify things that the human eye would struggle to identify, basically, just because it's so hard to grasp what is going on in these images. And what's interesting is you could think, AI for image analysis, there's going to be resistance from doctors because, we're, quote unquote, replacing them with the AI. And in digital pathology, that's really not at all the case. The reason being that pathologists are the first to know that there's a wealth of information in these images. So it's kind of like one of the pathologists summarized it by saying, they're asking me to describe what I see, but it's like I'm looking at this forest, this huge forest, and they're asking me what I see. So I can say I see, 3 oak trees and 2, bunny rabbits. But I can also say I see leaves that are more green to the left than they are to the right. Or I can say, the trees at the back seem slightly shorter than they are at the front. And they know that, they captured some elements of what they're looking at, but what they're looking at is just so much more rich. And up till today, they were the kind of almost the only ones who knew that they were looking at something that was just so complex. And so they've always been very, very keen to collaborate with us. And so we worked on basically capturing and collecting through the network of academic collaborations that we've developed at Owkin, digital pathology images and matching clinical outcomes to see to what extent we could build AI models to, for example, predict presence of certain biomarkers, likelihood of response to specific treatments, prognosis under specific treatments, etc. And we published several papers in kind of top journals to show that that AI was capable of bringing, I would say, value to all of these use cases. and we're still working. We still have a lot of ongoing collaborations to that end. And so then progressively, we decided to take some of our most promising AI models and develop them into diagnostics to be used in an actual care setting. And so that's how we created the diagnostics team, which I head, whose objective it was to kind of take the first research level models that we had developed on these images in collaboration with the pathologists and oncologists that we were working with, and making them into products that can be used by other pathologists across the world in other clinical care workflows. So that's the team that I head.
Awesome. So I have a few questions. It sounds incredible. And yeah, I've definitely learned a lot about the size of an image in pathology, which I definitely didn't know or appreciate before. So when you're looking at one of those big images, I think there's a way that I would think about this if I was trying to think, well, okay, I want to diagnose whether, I want to stage this tumor based on this slide. And you would have a predetermined group of characteristics, and you would train an AI to look at those characteristics and make a judgment and stage it. There's another way of thinking about this, which is there can also be lots of correlations that we're not even aware of that would actually point to the stage or the prognosis, or again, other things that we might not even know. And when you use the phrase research level model, is it that you're talking about models, algorithms, or even Owkin as a company, that you are looking for new correlations and trying to find new things and doing that research? Or is it that you're focused on building specific algorithms against a specific use case and using criteria and biomarkers and all the rest of it that we're already aware of? Like, where does that make sense? Am I making sense?
I think so. So
basically, are you looking for new correlations in what you're finding? Are you doing that research, or are you not? I guess is the simplified version.
Yeah. So the short answer is we are. So you could use the AI to do several things. You could use the AI to, for example, if we're talking about staging a tumor, to quantify, whatever features the pathologist is trained to look at that are built into the final tumor staging. So, some would call them like, human interpretable features. So basically features in the image.
Yes. Okay, interesting.
Human being, and you ask the AI to score them. So you say, how many lymphocytes do I have? How many, how many, blood vessels do I have? What's the level of necrosis? Quantify the amount of necrosis, and then take that and potentially make it into, a staging or something that a pathologist already can diagnose. Or you can say, and this was the approach that we developed at Owkin from the beginning, you can have a what they call in data science, a weakly supervised approach. So you say, I'm not going to tell you what to look at, or I'm not going to point you towards any feature. Just this is the slide and this is this stage. And you figure out what you need to figure out to make that link.
Interesting.
And basically the AI will analyze the image and learn to predict, for example, tumor staging without having to guide towards any specific feature. And what's interesting here is that the AI can not only identify features that, we as human beings have already identified as being important, but also can identify new features that potentially we hadn't known, had an impact on, for example, tumor staging. And I would say the level where this becomes super interesting is not, for example, to predict staging, but to predict overall, for example, prognosis. So you can go to a level finer of, instead of looking at 4 different stages, you can look at a continuous, for example, survival outcome and predict that directly from the images. And so we have use cases where we look at, for example, patient survival, and all we input is survival and the image. And then the AI kind of does its thing and looks at the images and makes its prediction. And so then we work with basically the pathologists that we collaborate with on, what we call like model interpretability. So understanding what the AI saw in the images that made it make its prediction. And so we have ways of basically then going back and looking at the model, pinpointing the areas that were deemed the most important by the model and making its prediction. And we then try to figure out if it's enriched in specific features that we either already know of or potential new features. So, for example, we're going to look at these areas deemed impactful by the AI and then have pathologists look at them, but in a blinded way, kind of like they look at, what was deemed important, what was not deemed important, but they don't know, that this is these categories are the categories of the AI. We just show them kind of two different sets of images, and they say, well, in this set of images, I see more necrosis. And so then we'll know that the AI picked up on a specific feature, and sometimes that feature is already known in the community as being important, and sometimes we identify new features. So this technology, in short, is basically key not only to make better diagnostics and better patient stratification, but then we can use it to circle back to basically more basic research to identify disease drivers and characteristics that are actually really driving these tumors.
Wow. This is incredible because I feel like I'm learning loads here. So if I've got this right, so the weekly supervised approach will mean that it's going to find these correlations. And my question was going to be, how transparent is it? So how clearly is it then going to tell you what it's using? And it sounds like incredibly so. So it's literally going to show you what it's using.
So we can't actually make it talk. At this point, we can't make it talk, human language and say, well, and I would say the strength of it is that it functions differently than the human eye and a human brain. So it can potentially see and do things that a human eye and a human brain can't do. It also sometimes can't do something that a human eye or a human brain can easily do. So it goes both ways. It's just a different way of processing information. So then that's where kind of our engineers have to get creative in, how to, as much as possible, down the line, build a bridge back to basically have a human interpretation of what is actually going on, and as much as possible understand and make sense of what the AI is seeing.
Is this relatively unique as an approach? Is this unique to you guys? Or is this what everyone's doing that talks about being in pathology and AI and imagery? Is this the standard approach? Where's your differentiation in this?
So the approach of really the weekly supervised approach and how we developed our weekly supervised approach, without being kind of the data science expert at Owkin, is one of our really key differentiators.
Yeah, 'cause it sounds it, yeah.
So at the time, a lot of companies, projects, research groups will work on extracting first features from the image. So, for example, taking an image and making it into kind of a set of histomic features, and then from those features running their calculations.
Yeah.
And Owkin was really one of the first ones to develop very strong methodologies for not doing that and taking directly the full image and inputting it into the AI. And then also building models that allow you to, because ultimately what the kind of data science work does is basically take these images and make them into numbers. And then that's what the actual predictions are made on. And how you do that, so how you take the image and make it into a set of numerical features, that's a very, very key step. So, what numerical features you choose to boil down your images to is, I would say, the most important step. And so this is called the feature extraction step. So the moment where you take an actual image and make it into a set of numbers. And this feature extraction step is something that also Owkin has worked extensively on, and that we have, patents, open access models, publications on really this one step of the data science work that basically turns an image into a set of numbers.
So fascinating. My next question is, based on all of this stuff, who is the customer? And I guess the reason I ask partly is to understand this as a business model, I think is really interesting for the space, because not only are you getting algorithms and answers when you think about we talked about staging a tumor can be based on these things that a human can see, and we can build an algorithm, all the rest of it. When you're adding the weekly supervised approach in, you're creating a lot of questions and answers. You're creating things that require a pathologist to then validate, and you're going to do that in a blinded study. And there's lots of work that creates too. And so at the end of the day, you're a commercial entity that needs to either create defensibility on those things like patents, all the rest of it once you discover them, and ultimately a product to sell. And so super interesting how you've managed to create a business model around all this stuff. So a bit of a broad question, but do you want to talk to me about the business side?
Yeah, with pleasure. So it's actually there are multiple kind of players in this business. I would say the first are pharmaceutical companies. So pharmaceutical companies have drugs that they need to target specific patients. So more and more in oncology, basically, therapies are targeted to specific subgroups of patients. And we rarely have a drug that comes on the market for all patients of a specific cancer. More and more, we have a drug that comes for a specific subgroup of patients. And the challenge for pharmas is at several levels. One, identifying the most relevant subgroup. So which are the patients that are actually going to benefit the most from this therapy? And so there's a kind of a development question of how to, better stratify patients. And so here we can use this AI that we have to identify the right subgroup of patients that respond, and basically better analyze the data to find which patients would most benefit from a specific therapy. And then how do you roll it out in practice? So that's the diagnostic part, which is once you do have a drug on the market, how do you, in a scalable way, identify the patients that can benefit from your drug? And one of the pain points that pharmas have today is that sometimes they have very powerful drugs, very efficient ones, but that only work on a specific subset of patients. And that subset of patients is difficult to identify at a diagnostic level. So, for example, there could be a molecular, sequencing-based test that is, for example, too expensive or too difficult to roll out at scale for patients to be systematically screened. So you'll have the drug that exists, the patients that are out there, but the match isn't being made, or the match is being made too late. And so I would say the beauty of pathology images is that they're systematically generated in all the care centers. They're always available. They're easy to process, and through their digital image, through AI, we can basically diagnose whether or not a patient is a good candidate for a drug. And so this solves kind of that very heavy pain point that we have today in matching the plethora of relevant drugs that exist with their right patients that are all very heterogeneous and all carrying different driver alterations, different molecular profiles. And so that's where the diagnostic really use case comes in, is really in helping making that match. And so where this becomes interesting for everybody is that for pharmas, this allows them to have drugs that meet their full target market. So to maximize the number of patients that can benefit from their therapies. For patients, this allows them to basically quickly be diagnosed with, whatever alteration or whatever molecular or a morphological profile that allows them to benefit from a relevant therapy. For oncologists, this gives them more options for their patients. And for pathologists, this allows them to extract even more information from the pathology slide. So it kind of brings keeps the pathology and brings the pathology image really at center stage of the cancer diagnosis story. So to give you an example, there are some drugs that'll target an alteration that's only present in 1 or 2% of all cancers. So you could say that's not a lot of patients, and it's okay if we miss them. But 1 or 2% times all the patients that exist, that's actually a lot of people. And some of these alterations are only, for example, today detectable through expensive genetic sequencing. So sometimes in research protocols in top academic centers, patients will be diagnosed, but most of the time they will never be tested for these alterations. And so that 1% of patients will never benefit from these therapies that could potentially really change things for them. And through digital pathology and AI, we can basically pre-screen patients. So just basically running the AI through these images, we can predict whether or not they're likely to be carrying these alterations. So this can basically flag for care providers or for pharmas, that are, for example, looking for patients to recruit in a clinical trial, that these patients are very likely to be carrying this special alteration. And, maybe you should take a closer look and get the sample, for example, sequenced to identify whether or not they are in fact carrying this alteration. And so that's kind of the idea behind the suite of products that we've developed on microsatellite instability, which is basically a biomarker that is predictive of response to immunotherapy. And we are able to, through analysis of the digital pathology image and AI, pre-screen whether or not these patients are carrying this alteration. So all this to say that in terms of business, this benefits kind of a lot of stakeholders, and in terms of capital gains, pharmas have a lot to gain from basically the development of these digital pathology and AI products. And so we're collaborating with them on developing some of these new products. And then ultimately, once these products are developed and launched, we're also talking to basically payers from the different countries that these products are rolled out in, to basically see how we can get reimbursement for these products and kind of what is the value that they bring to the overall kind of care workflow. So I would say it's a multi-step business model where we're going to work with kind of our strategic pharma collaborators to build the most relevant diagnostics, the ones that answer some of the kind of drug targeting and patient identification pain points that exist today, and the ones that we foresee will exist tomorrow as well. And then also downstream work with payers to figure out, what the economics behind these products are, and how can we get payment for them.
It's increasing specificity everywhere, isn't it? It's just making everything far more targeted and specific, and just decreasing waste. That's broadly it. And we know from the pharma side of things, as you quite rightly say, marginal gains in practical reality can be enormous sums of money, but also for those individual people that get the right treatment at the right time, literally life-defining, life-changing, life-saving. And so that's the whole point, that by increasing the specificity, you're reducing the waste. Therefore, people are more willing to put the money where it's actually going to work, or not more willing, they actually do put, the money and the resource and the product where they actually work. And I guess that's the whole point. I'm just interested from from all the work that you do in and around this, do you see this changing pathology in any kind of deep way? We talked at the beginning about the link between medicine and data. You could say medicine and technology and data. And I think there are some of these specialties that are intrinsically linked to technology, like radiology, like pathology. And that's where we've seen the initial first movers in AI, take hold and actually create impact and actually embed into the clinical pathway and this kind of thing. And I can see the relationship between what you're doing and the creation of new insights in pathology of what the human eye can't see and all those types of things. In the same way that maybe CT did or MRI did in radiology, it sort of changes the field and changes the way that the practice is done. Like, do you see a kind of, do you get what I'm saying? Like, do you see like a relationship where pathology is changed with this type of new technology coming in?
Yes. So the short answer, definitely. So basically, I would say, unlike radiology, pathology was, glass slide is still largely, on glass slides, so not digital.
It's a lot more analog, yeah.
Yeah, it's a lot more analog. And until, these AI use cases started coming out, the driver for digitizing pathology was not super strong. So we had basically potential telepathology, so the ability to send slides, digitally versus sending them out in, sending glass slides out through the post office. There was potential teaching purposes. So, for example, digitizing some, specific either representative use cases or rare use cases or interesting use cases, but that's still like a limited number of use cases that you're going to digitize. You're not going to digitize all your slides. And that was basically it. And then the value for everybody of digitizing pathology was not obvious. And then we saw these AI applications starting to come out on the first sets of digitized pathology images at a research level first. And I think that this opened up so many possibilities in everybody's mind. And then this basically, in a kind of chicken and egg kind of virtuous cycle way, started accelerating the digitization of pathology, which in turn increased the research capabilities and the amount of data science that we could do, which increased the proof points that we could do a lot with these images, which facilitated the investments on digitizing of pathology. So I think it's they're both kind of happening at the same time. And that's what's different from radiology. Radiology went digital, and then years later the AI came in. Whereas here it's going digital because the AI is here, and because the AI has such an impact on the ability to care for patients. So that's really what's driving this digitization, and what's going to ultimately allow the products that we're developing to have a very strong uptake. And so sometimes, when people ask, well, how do you know that these products are going to be adopted? Because not all labs are digitized. I really always tell this kind of chicken and egg story, that it's a very virtuous cycle where the more we develop these use cases, the more we prove that we can have an impact on patient outcomes through this AI, the more that drives the rationale for investing in fully digitizing pathology. And then I think for pathologists, this is really changing the way that they're considering, I would say, the future of their practice. Where they're moving away from the glass slide. They're working on fully digitized images, and more working on potential AI applications.
Yeah. Listen, it's absolutely fascinating stuff. I feel like I've learned so much today, Meriem. I really appreciate you coming on. Everything from the weekly supervised approach to finding new markers and changing pathology. I think it really does show for me the this budding symbiotic relationship between medicine and technology and data when we get it right. And I stress when we get it right, because going back to one of the first things that you said in your story, this listening first approach is so important, because if you don't do that, and if you don't understand naturally what a pathologist does in their day, good luck trying to digitize or add AI into literally anything that they do if you don't empathize and understand with the struggles that they have. And so I think it's a really nice story that you've told about knowing and understanding what actually happens on the clinical ground floor with technology and computers and data collection, to then actually now being a significant part of a company that's changing the way that we do that in a specific department and really advancing the field. It's honestly fascinating stuff. And yes, I'll be watching very closely to see what you guys do in the future. And I'm delighted to have had you on before you're particularly more famous than you are. So I see this being every success. I have to let you go because of time, but Meriem, before I do, for people that want to know more about you or want to get in touch with you, or indeed Owkin and want to learn more about Owkin, what's the best way for them to do so?
I think they can contact us through our website, or, if anybody wants to contact me, I'm also available on LinkedIn, for example. I'm always happy to talk to anybody if time allows it.
Awesome. Meriem, absolute pleasure. Thank you so much for joining us. And yes, I will certainly see you soon.